right. Well, thank you for having me back again. This is always a highlight of my year, and thank you to Bill Park and the whole Park family for hosting us. It's really a wonderful experience, and you guys are such an amazing interactive audience, which is always a pleasure. As Bill said, unfortunately, Dr. Scott Plotkin, who is our executive director at the Pappas Center for Neuro-Oncology, was supposed to join me, but he was unable because he came down with COVID, so I'm pitch hitting for you for kind of the main presentation. So I usually start off, the last couple of years I've started off showing you these slides just to provide a framework for what we are dealing with. And so you see on the top panel the 10 most common cancer diagnoses in the United States. And you'll see that brain cancer is not listed in that top 10 list. There are about 24,500 cases of primary brain tumors in the United States each year. But what you'll see is that it does make this list of the top 10 most common causes of cancer-related death. So unfortunately, brain tumors, while being rare, cause a very substantial high mortality rate. And you'll see that there are many different types of primary brain tumors, and the largest proportion of this is this group of glioblastomas. And unfortunately, glioblastomas are also the most aggressive type of primary brain tumor, and why we focus a lot on trying to treat glioblastoma. And so I thought I would start out by giving you some updates on some of the work we're doing at MGH and across the broader neuro-oncology community, highlights from the last year since we last met. And I'm going to talk a little bit about the CAR-T trial that we're doing. I'll talk a little bit about this phase 3 trial in a hard-to-treat midline tumor, and then I'll talk about a promising drug therapy for IDH mutant gliomas. And so starting with our CAR-T trial, this is an intraventricular CAR-T BiTE cells for patients with glioblastoma. And this came out of work from Dr. Brian Choi, who was our presenter last year. And CAR-T stands for chimeric antigen receptor T-cell therapy. And so what this means is we basically take a patient's own T cells, modify them so they attack a target on the tumor, and try to fight the tumor in that way. And so you'll see on the left, conventional CAR-Ts are engineered in such a way to target one specific antigen in the tumor itself. And what Brian was able to do is to create this bispecific T-cell engager, this BiTE, that targets a particular antigen, but then also secretes another molecule into the tumor microenvironment to target another antigen and to hopefully turn on the immune system so that it can destroy the tumor locally. And so this is how CAR-Ts are administered. As I said, we take out the patient's own T cells. We activate them, meaning we modify them so that they attack the antigens in the tumor. We expand them so we have more that we can then re-inject back into the patient. But the way we're doing it in this trial is we're actually injecting the T cells into the patient's brain, into the fluid-filled spaces that we all have in the brain. And the benefit of that is that it bypasses the blood-brain barrier, which does a really good job of excluding toxins from getting into the brain. So we don't have to deal with the blood-brain barrier. And so this is the status of the trial. We've completed our safety run-in cohort, which was mandated by the FDA. And so we enrolled 3 patients who received 1 infusion of the T cells, and then were followed for 30 days for any evidence of any toxicity or any side effects. And so those 3 patients have completed that toxicity window without any unexpected toxicities. And so we presented the safety data to the Data Safety Monitoring Board, and they've approved us moving forward. And we've submitted the paperwork to the FDA because we also need FDA clearance to move the trial forward. And we're hoping to get that in the next month or 2 so that we can open up the trial to more patients beginning of next year. So we're very excited about that. So the second update that I would talk about is this ACTION trial. And this came out of work by Dr. Isabel Arriaga, who actually came to the seminar a few years ago. And she had run a phase 2 trial using a drug in this very hard to treat tumor, which it's called a diffuse midline glioma with an H3K27M mutation. The details are less important, but in her phase 2 trial, you can see these are 2 different patients, 1 patient on the top panel, 1 patient on the bottom panel. And the uptake of the contrast dye here goes away significantly over time with this novel drug. And so that was a really promising finding. And so now she's co-leading a trial, a phase 3 trial, using this drug in this hard to treat tumor. And the reason I bring this up is because phase 3 is the next step before we get drugs approved. So it's really important for us in the brain tumor community to move drugs into phase 3 so that we can hopefully get them FDA approved. So we're moving the needle a little bit. And then the last update I thought I'd talk about was this clinical trial that was recently published in the New England Journal of Medicine of a drug called vorasidenib, which targets which was used in IDH mutant low-grade glioma patients. And what we found is that vorasidenib significantly improved progression-free survival compared with the placebo group. So it was 27.7 months versus 11 months. And so that was a really good improvement. And we're hoping that this drug will actually be reviewed by the FDA in the coming months and then hopefully actually get approval in the next year. So those are sort of my 3 updates from the year. And hopefully I've shown you really kind of the importance that we're moving more drugs through the system to try to getting them closer to FDA approval, which is really what we need. And so now I'm going to shift a little bit and talk a little bit about sort of the main presentation that Dr. Plotkin was going to talk about. And what we had asked him to talk about was really the concept of intratumoral heterogeneity and how we target and deal with intratumoral heterogeneity. So what we know about primary brain tumors, and particularly glioblastoma, is that these tumors are made up of multiple different subclones of cells that behave differently. So the purple cells, the teal cells, and the blue cells all can behave differently. And so this has an impact on how we manage and take care of these patients. So, how do we think about heterogeneity in tumors, and how do we manage that and incorporate that into the care of patients? We've actually learned a lot about heterogeneity in primary brain tumors in the last several years. This is a table from the World Health Organization classification of primary brain tumors, which was recently updated in 2021. And so what you'll see is a lot of tumors are now classified based on specific genetic mutations in those tumors. So not all glioblastoma is created equally. The different mutations that make up that glioblastoma can make it behave differently. And so this is another table from the WHO, and you can see here just sort of the list of different mutations that have been associated with individual brain tumors. And it can be as simple as a single mutation or many different mutations that characterize these different types of cells. And so when we're thinking about molecular mutations or this intratumoral heterogeneity, I always like to start with the positive since I'm an optimist. What is the promise of identifying these molecular mutations? And the real promise is, can we identify a driver mutation? Can we identify a particular change in the tumor that is what is causing that tumor cell to grow? Is it really just the purple cells that are growing and the teal and the blue cells are not really contributing to tumor growth? If we can identify that target mutation, or that driver mutation, then targeted therapy affecting that driver mutation can be helpful. And so I already gave you an example of this. We talked about that vorasidenib trial with the IDH mutant tumors. And so by targeting this driver mutation with appropriate drug, we are now having an impact on the disease. The other place we've made some progress is there are some tumors that are driven by this BRAF mutation, and about 5% of glioblastomas have a BRAF mutation. It's a pretty small number, but we are seeing responses to BRAF, targeting that BRAF mutation in gliomas. And so this is a patient of mine who you can see the tumor here in white, and 12 months later, you really don't see any active tumor. So again, if we can identify that driver mutation, then there's some hope there. I think the challenge that we face in our world is that resistance eventually arises, and this usually happens because one or more alternative subclones starts to grow. And so the blue cell was maybe not doing much before, but now it's starting to repopulate and take over the tumor and cause the tumor to grow. And so what we really need to do to understand these alternative resistance mechanisms is to get tumor tissue, to look at them under the microscope and really figure out why that blue clone is suddenly starting to grow again. Unfortunately, it's very hard in brain tumor patients to kind of go in and do repeated brain biopsies to get that tumor tissue, to look at it under the microscope and figure out what those resistance mechanisms are. We're trying to be a little bit more aggressive to do that because we understand the importance of identifying this, because that's what we need to design the next therapy or next combination therapies. So, where is the challenge with these molecular mutations and intratumoral heterogeneity? One of the big challenges is, what if there is no single driver mutation? What if there are multiple drivers, multiple cells that are growing, or there may be an unknown driver mutation? We haven't quite identified what is making those cells replicate and grow. So what are some of the approaches we are using to try to tackle that? And one of them is using immune approaches, and the other is looking at clinical trial optimization in these platform trials. And so when we think about those immune approaches, you've seen this slide already tonight, because this is one of the advantages of the CAR T approach that we talked about. We're targeting more than one antigen in the tumor, trying to knock out more than one cell type. And then, as I said, this bispecific CAR T cell, what it does is it's supposed to turn on the immune system locally within the cancer and bring in more immune cells, more T cells, that then can start to recognize other antigens, other abnormalities in the tumor itself. Maybe not necessarily the one that we had identified or engineered our cells to, but it continues to recognize other subclones as foreign and attacks those. And then the last way we're doing this is with clinical trial optimization and this concept of platform trials. And so this is a platform trial that is ongoing. We have open at MGH. It's called the INSIGHT trial. The H is supposed to be lowercase. And so basically what it does is enrolls patients with newly diagnosed glioblastoma. And what they do is they collect the tumor tissue and they sequence it, looking at the different mutations in the tumor. And then patients are randomized adaptively into, at the start of this trial, it was one of 4 arms. They had a standard of care arm or one of 4 potential treatment arms. And then patients were followed, looking for how well they did, how long the tumor was controlled, progression-free survival, as well as how long the patient survived. And so as the patients were enrolled on each of these arms, they could figure out which arms were working and which ones weren't. So for example, the CC115 arm wasn't doing as well. Those patients enrolled on that arm were not doing as well. So that information fed back in to the randomization algorithm. And so fewer patients were randomized to the CC115 arm and more were randomized to the other more promising arms. And so the results of this have been preliminarily published. Oh, and I should also say, as new drugs come on the market or come to potential be effective in brain tumors, they can start to be added in as additional arms of the trial, and they just keep getting added in as new arms, as new drugs become available. And so the preliminary results of this study were published, and so what they found was the abemaciclib and the neratinib arm improved progression-free survival compared to the control, but unfortunately no arm improved survival. So we're going to keep working on this and open up more arms. But this is the benefit of the adaptive randomization. So this sort of graphically shows that benefit, where what they would have, if you had randomized patients, one patient to each arm, what they would have enrolled, more than 15 patients to that ineffective arm. But with adaptive randomization, they were able to minimize the number of patients that were enrolled in the less effective arm. So you're really enrolling fewer patients to a less effective treatment. And then I'll give you just one last example of these platform trials. This one is called the Adaptive Global Innovative Learning Trial Design, or the AGILE trial. It's the same idea where patients with glioblastoma get enrolled. Now in this trial, what they're doing is they're looking at the tumor tissue that was taken out at the time of surgery, And they're trying to look, they have some a priori hypothesis that certain mutations will respond to certain drugs. And so based on that, they will enroll patients on certain arms of the trial. And then patients will get followed over time. And then, as I said, we don't necessarily know what are the driver mutations, why somebody may be responding to a drug or not. And so as they follow patients, they will go back and look at the tissue that they had and understand what the changes are in the tumor and be able to figure out if they can identify new changes that are suggesting that somebody's responding to a particular type of treatment or not. And then they can selectively, in their randomization model, start to shuttle more patients who have that biomarker into the arm where they may most likely be benefiting. And so it's incorporating biomarkers both in the randomization process and when assessing the response to the drug. This slide is meant to overwhelm you. You're not really supposed to pay too much attention to it. It's really just to highlight that these are great trials. These platform trials are really, I think, a very big boon to how we're going to do clinical trial and drug development in glioblastoma going forward. But they've really increased the complexity of how we run clinical trials. You're basically running 4 or 5 clinical trials within the same kind of umbrella trial. So it creates a lot of infrastructure and a lot of work for our research team. But I think it's really exciting. So kind of to summarize, these platform trials, they really allow simultaneous evaluation of multiple drugs. They minimize the number of patients randomized to standard of care treatment. A lot of brain tumor patients who come to MGH really, they recognize it's a really difficult, challenging disease. They want that novel therapy. They want that, interesting investigational drug. And so we minimize the number of patients who are just getting standard of care. But at the same time, wearing my scientific hat, I need that standard of care backbone so I can really prove whether or not an investigational drug is better than the standard treatment. And then they also incorporate this adaptive randomization. And adaptive randomization, as I said, can be biomarker driven based on that intertumoral heterogeneity, the molecular makeup of the tumor. And then it also can be, the randomization can be adjusted based on information that's learned during the trial. How effective is a drug, or can we identify particular biomarkers that suggest somebody's going to respond to to certain drugs that are in the trial. So in conclusion, we've spent, the last 15 minutes or so talking about intertumoral heterogeneity, which really is a big challenge in neuro-oncology, but it's also been a big benefit as well, because as we understand the molecular makeup of tumors, it really has improved our tumor classification. So we better understand these tumors and what's driving the biology of them. And if we can identify driver mutations that can be targeted, that is a great step forward in caring for our patients. And really, what we need to do is design therapies that can adapt to an evolving tumor. These tumors are very smart, and they succumb to Darwinian pressure, and so they start to develop new pathways forward. And so we need to figure out ways of turning off those other pathways. And one of them is potentially to use the immune system, which is our best native way of fighting foreign antigens. And then the last thing we've been doing to tackle intertumor heterogeneity is optimizing this clinical trial design. So we really can learn the most we can about novel drugs and minimize patient exposure to ineffective therapies. And I think that's it. Thank you to all of you for coming. Sorry,
We just heard now. Let me remind you, if I may, that we use this portable mic not so you can hear it in the room, but so that we can record it for the video that we play on milesandmary.com. And Bill's going to be passing the mic, and John will start with you. Here you go.
What's a typical time span for beginning and ending these experiments? In other words, when you get a patient comes in from day one to how long does it take to get a result?
To do the biomarker testing?
Well, whichever place you're trying to get to, is it vary from patient to patient, or let's talk about your most optimistic result. How much time do you spend with one patient?
Do you mean to get the results of the clinical trial or to get the results of the testing off of the molecular?
I would say the latter.
To get the molecular testing. So this is, it's changed, and it's really become a bit of a challenge, because I used to be able to say, it's about 5 to 7 business days from when a patient has surgery to when I can give them the diagnosis. But that has changed now in this molecular era, because they have to do more stains, more testing. There's next generation sequencing that's done. So sometimes it can take a little bit longer, anywhere from 10 to 14 days sometimes before you can give a patient a full answer as to what they're dealing with, which obviously from a patient perspective is not ideal.
Ralph?
In these different trials, how many people are typically in a trial?
So it's usually for these trials, it's anywhere from 50 to 60 people in each arm of the trial. And so, so it's a fair number. And so that's why most of them are multi-center. So the Agile trial is actually an international trial, and Insight is it's within the United States, but multi-center.
That seemed like a lot of people.
Right.
Yeah.
In these randomized trials, so people don't know, they agree to be in one, you don't know where you're going to land. But let's say you fall into the pile that is doing less well, that others are being diverted from. Are you told of that, or do you have an option to then move from that position?
So we find once the results are published, so that gets fed back to me as an investigator. And then if somebody is still on that arm that's proven to be less effective, then they have the option of either continuing or switching to a different therapy.
So when you talked about the global initiatives that are going on, the new
Agile.
platform, that's reminds me a little bit like open source, like everyone around the world is sort of like sharing data into one, starting to, or at least some of your researchers around the world are all with their patients going into one data for you. So as a scientist, you can see it all coming together, or is it moving in that direction, or you hope maybe it does, but right now it's...
So it's still in the context of a clinical trial.
Okay.
So you have to open the clinical trial at your individual center where you're seeing brain tumor patients.
So Mass General can't, if I'm in Switzerland and I have glioblastoma, we're not connected to Mass General at this stage.
Right.
Yes,
But there may
okay.
be, the Agile trial may be open someplace in Switzerland.
Right, but it's got to be a connection.
Yes,
It
through
just
the
seems
trial.
promising the more you can connect. more
Right. And there's different
patients.
practices around the world. And
Yeah.
so it's a good opportunity to sort of see how different treatments play out in different different parts of the world.
Thank you.
I have two questions. One, is there an average age where people get glioblastoma? And also, with AI, there's a lot of talk about AI helping in science these days, and I wondered if there was any possibility of you advancing more quickly with AI?
So the average age of onset is about 64. It unfortunately hits people right when they're getting ready to retire and enjoy their well-earned retirement. And then AI is definitely coming into play. We're using it. So my lab does a little bit of deep learning and AI stuff. We're using it more in less in diagnosis, but more in looking at tumor response to treatment. Can we figure out, just based on imaging, can we predict who's going to respond or not respond? Can we outline the tumors so we can get a more accurate response rate, because we measure the size of the tumor as our determination of if the tumor is responding. And so can we use these deep learning based segmenters to outline the tumor and get a better assessment of response? I suspect, AI is doing some really interesting stuff where it can look into medical records and it can predict things like depression ahead of time before it really gets noticed by, say, a primary care physician. So you can imagine maybe it'll be able to pick up signs and symptoms before it kind of comes to a head.
You talked about the arms and if there's a unfavorable arm, kind of on your question. And then you put less people in the unfavorable arm. And do you not give up on that arm because you want to make sure it's not a fluke that it's unfavorable, like scientific method wise?
Exactly.
And then if somebody is told that they're in the unfavorable arm, why would they continue?
Because some people may be having a response, and it might just be there's something about their tumor that is sensitive to that drug. And so they may think that they are benefiting from it. You don't want to take away a treatment from a patient if they feel like they're benefiting from it. Because some people just feel like this is the drug that is keeping my tumor under control. It goes to that heterogeneity of the tumor. There may be some biomarker in that person's tumor that we haven't identified that is responding to the tumor, but it's such a small subset, it's not being pulled out in the kind of larger group when everybody's averaged out.
Yes, when you showed the slide that in the beginning that unfortunately that very high mortality rate, my question is that drug that begins with V, how that was really extending life. I personally think living in this area that MGH, has such a high reputation, does the fact that there's that high mortality rate, but that drug beginning with V is doing pretty well in trials, do you have any pull with the FDA to fast track?
So I think it is getting fast tracked. And so it should hopefully be approved. I can't imagine that it won't be, but I can't speak for the FDA. But I think another issue, and maybe this is getting into the weeds a little bit, is sometimes with these smaller drug companies, they need to rev up manufacturing as well. And they need to be able to make enough drug to disperse it to the population at large. And so part of it is FDA approval, but the other is just the logistics of having enough drug manufactured.
How are the drugs being delivered? The blood-brain barrier and so on. I mean, is CAR-T being used or?
So the CAR-T we're doing directly into the brain to bypass the blood-brain barrier.
Right, but it's delivering a drug that is seeking an antigen that then is going to be killed.
Right.
And then it's also, with the BiTE trying to stimulate the T cells to kind of
Turn on and wake
really
up.
go, which
Yeah.
is the being done in other diseases, lymphoma, myeloma,
Right, right.
even breast cancer.
And all of those can be given by vein because they don't have to deal with the blood-brain barrier.
Yeah.
But in brain tumors, by giving it directly into those fluid-filled spaces, the cerebrospinal fluid, and we don't quite know. This is, I do a lot of neuroimaging, and there's a very interesting neuroimaging approach to sort of maybe look at CSF flow through the brain. And so I'd love to add that into our trial to see if we can see kind of the flow of CSF from the ventricles, from the fluid-filled space into the brain. And so can we see how the T cells are tracking? Because we have to get it into the brain substance itself. And we don't necessarily fully understand how that happens. But at least it doesn't have to go through the blood-brain barrier.
How are these drugs developed? What's the initial basis, and what's the relationship between you or Mass General and the drug companies? How does this all get evolved?
So for the CAR Ts, those came out of a lab at MGH that Brian Choi was in, and now he started his own lab, and so he's working on creating new CAR Ts. And so they're developed locally, and then the hope is if our trial is successful, maybe we can license it to a drug company to kind of develop further.
You can correct me if I'm wrong, but my impression is that if you attack cancer mechanically with surgery or targeted radiation or some other basically mechanical method, you always have the risk that you're going to miss some of it and it can regenerate. It seems to me the advantage of a chemical treatment is that if it's effective, it theoretically is going to get all of it. So I'm intrigued with you kept referring to the adaptive response of the tissue and that it could somehow reformulate itself and not, be killed by the chemical. What I'm curious about is the timeline for that to happen. If you truly have an effective toxic chemical, shouldn't you be able to just knock it out before it has a chance to adapt and regenerate? And how do you condense that timeline to make that happen?
Yep. Well, so part of the role of surgery is to debulk the tumor, and so you minimize the number of cells that are left behind. So you have less tumor cells that can maybe go haywire down the road. And so it is a good starting point if you can take out the bulk of the tumor and then you zap it with radiation, because we know that's effective. And then we combine that with drug therapy. But the challenge is, these cancer cells are just so smart. And yes, ideally, if we could have a drug that was 100% effective and eradicated every single cancer cell in the brain, then we can cure brain cancer. We haven't found that drug yet because these cells have really, they'll stay quiet for a certain amount of time, and that's very variable patient to patient. And then something just causes that one cell to wake up again and no longer be sensitive to that drug that you've been giving the patient. It's oftentimes just a bypass mechanism. So you've blocked the pathway that's telling the tumor cell to grow, and so it stopped growing, but then a bypass pathway gets revved up, and so it just starts to replicate again. So yes, it would be ideal, but we'll get there.
Okay. Maybe just one last one. Over the years, we've talked with you about cooperation amongst the community entirely. And it seems like this platform clinical trial design sort of demands cooperation. And now we're talking about internationally in hospitals that aren't allied at all. Is it getting a little better?
I think so. I think there's recognition that with these increasing subsets of tumors, we have to do these larger trials where everybody participates to put together these, subsets of tumors that are, glioblastomas, as I said, is not all glioblastoma. It's these increasingly small subsets of tumors that are labeled glioblastoma. And the only way we're going to be able to do that is to participate in these larger, multi-center trials so we can put together enough of those subset glioblastomas to come up with the best answer for patients.
It sounds good. Okay.